ASO steric blockers for exon inclusion have a different design from ASO gapmers, but they use some of the same chemical modifications. So with gapmers, we have this high affinity methoxyethyl modified sugar on the ends, and then we have a DNA gap in the middle that recruits RNase H and allows it to cut this RNA, and that way we can get rid of disease-associated RNAs to treat some diseases. Now, if we design the oligonucleotide instead using the methoxyethyl sugar throughout the length of the ASO, now there's no gap. It doesn't recruit RNase H, but what this will do instead is to prevent protein binding to this region of the RNA if it's binding tightly enough. And of course, this is not a catalytic process, it's reversible. This can go on and off with the binding, and so therefore there are going to be less off-target effects than gapmers. There's nothing irreversible that's happening. So how can you use this type of a steric blocking ASO? We're going to be talking mostly about altering pre-mRNA splicing. Right now I'm going to talk about using this for exon inclusion by blocking an inhibitory factor, and then we'll come back to using this type of design for exon exclusion by blocking positive regulatory factors for pre-mRNA splicing. But there's a lot of other potential applications too. Some diseases like myotonic dystrophy are associated with toxic RNAs that bind proteins and disrupt cellular function. If you design a steric blocking ASO to those toxic regions, you may be able to treat those diseases by reversing the effects. These are being used to prevent nonsense-mediated decay of RNAs in some diseases, and this can increase protein production. As well, there are regulatory RNAs, microRNAs, that can suppress gene expression. We can target these regulatory RNAs with steric blockers, or we can target their binding sites in mRNAs in order to upregulate gene expression. Many genes have upstream open reading frames that can be targeted to induce expression or increase expression of the primary open reading frame. Another example is modulating mRNA stability through targeting poly-A signals. Once we have a design like this, there will be all kinds of ways that we think of to use this. But I'm going to talk about using that design to treat a specific disease called spinal muscular atrophy. Spinal muscular atrophy is the leading genetic cause of death in infants, and it results from a defect in a gene called survival of motor neuron. In normal individuals, this gene has 8 exons that are spliced into the mature mRNA, but in patients with spinal muscular atrophy, they've inherited a defective copy of the gene from both of their parents. It is a recessive disease, and if they cannot make this protein, their motor neurons don't survive and they die. There are several different forms of the disease. This gene is unique, more or less, in that there's a backup copy of the gene, an extra copy in the genome called SMN2, which is almost identical to SMN1, except for a C to T change in exon 7. This single base change leads to the binding of a protein that inhibits the normal splicing of exon 7, so that it's excluded from the mature mRNA in 90% of the RNAs that are made. However, using a steric blocking ASO designed to bind to the site where that inhibitory protein binds, you can prevent that suppression of exon 7 splicing and have it included in 90% of the mature RNA, thus restoring production of functional SMN protein. Now, this sounds like a great idea and works in cell models. Is it going to work in humans? One of the limitations is that because this is in neurons, in order for the ASO to get to the neurons, the ASO has to be given by intrathecal dosing. That's lumbar puncture. And this ASO has a fairly long half-life, so once you've given a few loading doses, that only requires 3 doses a year. Clinical trials of this approach started in 2011, and at first it wasn't clear whether it was working or not. The FDA wanted to see a randomized controlled trial, and so that was performed in infants with the most severe form of the disease, where the controls by 1 year mostly have died. And the nusinersen treated patients very clearly had improved survival. This is looking at survival, including whether or not the patient requires ventilation, because the normal course of the disease for kids is they can no longer breathe on their own. And this is looking at overall survival. And very clearly, there was a statistically significant benefit to nusinersen. The FDA approved it. And this was the first treatment for the cause of SMA. It radically changed the disease course, has saved many lives. And as time has gone on, it's been approved for the milder forms of spinal muscular atrophy that are not rapidly fatal, like later onset spinal muscular atrophy. Here, instead of looking at survival, you can look at function. Are the kids meeting the normal developmental points along the way? And there are scales for their developmental function, which, without going into the details, you can see the nusinersen treated kids are going up in their function. They're gaining functional abilities, whereas the children who were randomized to the control group lost function or simply did not improve. So this is a dramatic change in the disease course. This shows the potential power of steric blocking ASOs to treat very severe diseases. Spinal muscular atrophy is somewhat unique in having that backup copy of the gene, but there are many other diseases as well where this type of design can benefit patients.